Am I audible? Yeah,
Yes, sir. Yes, sir.
here. Hello.
Hello.
Good morning.
Yes, sir. You are audible. Please go ahead. Yeah. Please go
Okay,
ahead, sir.
sure. Thank you. Welcome back. Um, just we had a wonderful morning session with a keynote by Dr. Shobha Ranganathan and also followed by a very enlightening panel discussion. So now we start with our session, the first session. Before I start, I'd just like to thank Devendra and Prashant for giving me this honor. And I think my co-chair will be joining shortly. Um, so we have this first session on the of INVIX 20 on advances in eukaryotic genomics. We have three invited talks in this, by one by Dr. Debashish, and another by Dr. Abhishek, and the third one by Dr. Prashant. And there are two oral talks also, and followed by a poster talk. Without wasting much time, let me introduce Dr. Debashish. Um, So Dr. Mohanty is presently a staff scientist in Bioinformatics Center of National Institute of Immunology, New Delhi, India. He is known for his studies on structure and function prediction of proteins, genome analysis, and computer simulation of biomolecular systems. Dr. Mohanty is an elected fellow of all the three major Indian science academies, that is Indian Academy of Sciences and the Indian National Science Academy and the National Academy of Sciences, India. The Department of Biotechnology of Government of India awarded him a National Bioscience Award for career development, one of the highest Indian science awards for his contributions to bioscience in the year 2009. He has published more than 100 articles in nationally and internationally reputed journals with an H index of 27. Dr. Mohanty studied his MSc from Indian Institute of Technology, Kanpur, and then did his PhD from Indian Institute of Science, Bangalore. He is also the alma mater of Hebrew University of Jerusalem and also the Scripps Research Institute. He is awardee of several acclaimed science awards like 2005 OBA Samantha Chandrasekhar Award and 2007 Rajiv Goel Prize and many more. It's my pleasure to invite Dr. Debashish to present his talk for today. Dr. Debashish, please. You have to unmute. You have to unmute, I think.
Thank you. Opportunity to come and talk in this conference. So what I am going to talk today is machine learning based approach for deciphering metabolites which mediate interactions between host and the microbiome. What I'm trying to say is the microbiome is a term that describes the genome of all microorganisms living on and in a host, and it affects the many of the host processes as well associated with health and disease in many different ways, like biosynthesis of vitamins influences immune maturation, metabolism of branched chain amino acid, and the number of genes in the microbiome is several millions. This is a recent study which estimated how many genes could there is. So in addition to our host genes, we should also be bothered about how these genes from the microbiome are affecting the host processes. So what I would concentrate specifically in this talk is how secondary metabolites which are either derived or originate from the microbiota, or the host metabolites which are transformed, or xenobiotics which are transformed into metabolites, how they affect different processes. So in this slide shows many different metabolites which comes from microbiota associated with the stomach, intestinal tract, skin, oral cavity and things so on and so forth. So the broadly, so all these studies, what has been done till now would broadly point out that these type of host associated, this microbiome associated metabolites can be grouped into three different types, like the metabolites which are de novo derived by synthesized by the microbe. So these are mostly bacteriocins, or what are called REAPs, secondary metabolites like polyketides, non-ribosomal peptides. And there are another class of metabolites where a host-derived metabolites is transformed to a different metabolites by the microbiota or enzyme coming from the microbiome, or genobiotic, that is a drug or a diet metabolite, is transformed. So these are few examples of how this sort of a transformation is working. Like, let us say in this mycobacteria called C. scindens is contributing a gene or a enzyme which is converting a primary bile acid to secondary bile acid. And if there is a secondary bile acid is there, so now in the Clostridium difficile, so this is inhibiting the growth of vegetative growth of this and production of toxins. Also, if there is a dysbiosis between these two microorganisms, so then that leads to a disease situation. That is what is shown in this. So the key to that is a microbiome derived enzyme which is converting primary bile acid to secondary bile acid. Same way, this is how the enzyme which is coming from E. lenta, that is again one of the microbe, is converting digoxin, which is a cardiac drug, to the inactive form. So now, if those microbiota is higher in the patient, so then this drug is not working. So this is also another example. And these are a few other examples of, let us say, they are making a molecule called lactocillin. It is coming from a bacteria which is there in our microbiome, which is Lactobacillus gasseri. And what it is inhibiting is the growth of other pathogens like SRES and things like that. This is another example called lugdinin. It is coming from Staphylococcus lugdiniensis, and it is preventing the growth of Staph aureus. So what my talk would concentrate is how you can analyze the microbial genomes and understand or have a computational method where you can predict which are these metabolites which are biosynthesized by the genomes of the microbes. And that I would primarily concentrate from the three different types, only the ones which are the de novo metabolites, that these metabolites are biosynthesized from the microbes. The other two classes I will not talk here. So now the information to make any of these metabolites, secondary metabolites, whether it is a polyketide or a non-ribosomal peptide or a class of molecules called REAPs. What REAPs stand for is ribosomally synthesized post-translationally modified peptide. So the information is there in the genome in the form of key catalytic domains, their 3D structure, and using that, a series of reactions are catalyzed, and finally these metabolites are being made. So now, if you have the genome sequence, the complete genome or a metagenome sequence of the microbiota, we can analyze, and by a computational tool, we can predict what sort of the molecules being made. That is what is the purpose of the talk. And you may ask, has it really been feasible in some place or not? So there are many examples. Here I am giving you an example called informatipeptine. So the reason it was made informatipeptine, this is a molecule which is discovered using an informatics approach by identifying the genome and predicting a gene cluster and showing that it is making this not particular REAP, ribosomally synthesized post-translationally modified peptide. This is a class of lantipeptide, and this was subsequently confirmed by mass spectroscopy. So there are many such studies which shows that by genome mining, that means by analyzing the genome, you can really predict secondary metabolites. And this is, and there are the few class of metabolites which you would concentrate here in this talk is a class called polyketides, which are made by a class of enzymes called polyketide synthase, like erythromycin is an example. And here there are many catalytic domains, and what this color coding is shown is like ATACP. These are doing this moiety. This KSAT, KRACP, this is adding the moiety which is colored in yellow. So by a combined assembly line mechanism, you are getting all the moieties, they are joined and cyclized, you are getting erythromycin. So there is another class of enzymes called non-ribosomal peptide synthase. So they are making peptides, but they are not made in the ribosome. So they are made using a protein templates, and there are catalytic domains which are specific for amino acids. Something will add phenylalanine, another enzyme will add asparagine, another enzyme will add tyrosine. So you have a peptide and there is a thioesterase which is going to cyclize it, and you are getting this cyclic molecule, let us say tyrosidine A. So now in the same way, there is another class of molecules which are called ribosomally synthesized post-translationally modified peptides. So that means this is a small peptide which is synthesized in the ribosome, what is shown here in green, and in its genomic neighborhood there are other modifying enzymes, and these modifying enzymes modify the peptides, install many different types of cross-links. So these are mostly bacteriocins, glycosins, sactipeptide, lanthipeptides. This is the class of these metabolites. So what I am going to tell you today, how you can analyze the genome and predict these classes. And what is shown here in more detail is the type of cross-link these enzymes are putting. As you can say, what we start from a simple peptide, which we can infer from the genome, and we have to tell which amino acids are modified and how these are cross-links. And can you really predict, because this can be cross-linked in many different ways, can you really predict which cross-link the nature prefers? And this is what, and there are many tools which were doing this, like anti-SMAS is one tool. It can analyze the genome, but what it can predict is a linear moiety, but how it cyclizes, it cannot be predicted. So PRISM is another program which can again predict linear moiety, but it enumerates all possible cyclization patterns. That means it is going to give you a large number of possible cyclization patterns. It is not going to tell you which type of cyclization is preferred. And this is another program called SBSPK, Structure-Based Analysis of Polypeptide Synthase and Non-Ribosomal Peptide Synthase. This is a program which has been developed in our group. This could also earlier predict only linear precursor. So now the question is, how do you go from linear precursor of the moiety, which you are deriving from a bioinformatics analysis, to predict the correct cyclization pattern? So this is where the machine learning is coming to help us, and you must have heard recently the news how machine learning has successful in predicting the protein structure during the CASP context. And what is shown here is the output of a program, or the result of the program called AlphaFold 2. What everybody is excited that it is able to predict protein structure as good as experiments. These are the superposition of the predicted this. And before this, people had used machine learning to tell how to address questions in chemistry, how you are able to tell the synthetic route of a molecule, which are the reaction steps one should, how do you synthesize these molecules. This is what chemists do. So a AI program which is trained on various reaction mechanism is able to predict it. So the potential machine learning application for bioinformatics and chemoinformatics, if you join them together, you are in a position to address the questions which I highlighted. So let us say this is shown here, the precursor molecule before cyclization to generate erythromycin. So using a bioinformatics approach, by taking which catalytic domains are there, which moieties will be joined together, you could predict a linear structure, and this is indeed possible, and we have many publications from our group and other groups which have shown this. But now what I would explain you today, how do you predict, if you theoretically cyclize this carbonyl oxygen can cyclize with any of the hydroxyl group, you will generate all these 6 possible structures. But you have to tell which of these 6 structure is the correct structure. That means this one is the correct cyclization, these are all incorrect cyclization pattern. So if you think in terms of machine learning language, this correct structure, you can say this is the positive data, the incorrect structure is the negative data. So if you use this data to train a machine learning classifier, you can indeed predict this cyclization pattern. So, and once you do this, so you need, of course, as an example, many known cyclization pattern, that is going to be your training data. You are training your algorithm on this, and then you are evaluating how good is the algorithm and this. And what this training process does is, the data might be originally very complex, that means positive and negative data is mixed up. It is thinking of a mathematical transformation which is able to separate the positive data and the negative data, so that then it becomes easy to distinguish. And once you do that, And how good is your prediction is decided based on this graph, what is called a ROC curve. It plots at a false positive rate on one axis and true positive rate on other axis. If your prediction is 100% perfect, you will get a rectangle, and the area on that means at a 0 false positive, 100% true positive. So that is going to give you area of 1. But any other prediction could be, and if you do a random prediction, you will be get it along the diagonal. That means AUC value will be, area under the curve will be 0.5. So anything between 0.5 and 1 is a good prediction. So the more convex is the curve, that is better. That means at a less false positive, you are getting more true positive. So, but to do this, convert these chemical structures into a machine readable form where machine learning can work, you need to do what is called a encoding in terms of a feature vector. So in the drug industry, there are many type of chemical fingerprinting is being used. So what we used, what is called a Morgan circular fingerprint. So what it is keeping track is which atoms are there, how they are bonded, is it a double bond or single bond, and things like that. Using this criteria, it is converting a chemical structure into a vector string of zeros and ones. So these zeros and one are essentially representing all these chemical structures. Once you do this feature encoding, you can use any machine learning classifier. So because the time is limited, I would just tell we have used one type of classifier which are called random forest classifiers. And the way this feature encoding is done is you center on every atom, you draw a circle of different radius and try to check which atoms are present, how it is bonded, and as you keep moving these atoms over the entire molecule, you generate the entire fingerprint. So once you do that, so we did this exercise and we used a data set of polyketides and non-ribosomal peptides for which we knew the correct structure and the correct cyclization pattern. And as in any machine learning, this data set will be divided into positive and negative data set and trend. And this is the performance of the machine learning classifier. That means if you look at this curve at a 20% false positive, you are going to do a true positive prediction of 80%. And so under different conditions, the performance is reducing. I will not go into the details, but if you see the sensitivity, specificity, precision, and all these statistical parameters, all these data goes on to show that we are able to predict the cyclization pattern with a reasonable accuracy. So what this table shows is if somebody has the correct linear structure, you are able to predict the cyclization pattern with 95% accuracy. Whereas if you have a linear structure generated by a software like Antismas or SBSPKS or Prism, the predicted structure, the performance is reducing. And that is what has happened. And so once we did that, so this is what I told as of now is summarized here. So you started from the genome, you predicted what are the gene clusters present. In gene clusters, what are the catalytic domains, and then from that you predicted the linear structure, and now you used machine learning at this step to go from the linear structure to the cyclized structure. And this work now, that is what I just told you, this has been recently published from our group in bioinformatics. And what it is telling you is starting from a linear structure, from a genome, you can tell how many gene clusters are there. For each gene cluster, you can predict which are the secondary structures of the metabolites. And of course, the predict up to the reasonable accuracy, which I showed before. And now these are for polyketides and non-ribosomal peptides. In the same way, if I want to apply this problem called RIPS, as I told you, RIPS are ribosomally synthesized post-translation modified. So here you have to predict these lanthionine bridges. That means cross-links between different residues. Theoretically, if you generate cross-links, these are the all possible cross-links. But the true cross-links are the ones in blue, and the red ones are the false or wrong cross-links. So again, use the same trick. Try to use the blue ones from the known data as the positive data, and the red ones which you will generate theoretically as the negative data. Train a machine learning classifier, then you are in a position to predict this also. And this also we have shown in our earlier work that you are able to predict the cyclization patterns in RIPS. And the overall method, the flowchart is given in this graph. I will skip this, and what I would try to do is now if we apply this to a data set of 15-20 genomes, the complete genome sequences which has come from gut microbiome, so in each of them we could find the different classes of number of lanthipeptides, glycosines, lysopeptides, like how many different types of RIPs are there. For some of these RIPs, we could predict the cross-linked chemical structure, and we can compare with the known structure. This is a molecule which clustering structure we predicted by using our program RIP miner. This is a molecule which is similar to this a known molecule called nisin U up to 99%. And this chemical structure similarity is measured using a thing called what is called Tanimoto coefficient. So now the same approach, like now this RIP miner, if you give a genome, like we tested another example, there was a paper published where it said an antibiotic which is coming from a microbiota is the restoring resistance against vancomycin resistant enterococcus. It is a Glaucia producta. We took the Glaucia producta genome and gave it to the RIP miner program, and it predicted this chemical structure. And if you compare it with the actual structure which has been experimentally characterized, if you find that all the crosslinks have been correctly predicted except for this threonine, which sequence is like where it is the software doesn't predict it is to be a DHB, which is a modified residue, which is actually modified in the experiment structure. So this tells you the accuracy to which you can predict these chemical structures. So this is where I will stop, and the overall conclusion from my talk is a evolutionary analysis of known natural product biosynthetic pathways in combination with protein structure modeling can help in developing predictive rules for correlating sequence of secondary metabolite gene clusters to their chemical structure. And there are these are the few things I did not cover in more detail. And combining bioinformatics, computational chemistry, and machine learning approaches can help in discovery of new biosynthetic pathways by genome mining. And these are the people who have contributed to the work, and the primary work what I presented is done by Priyesh and Sana Amir, and these are the other people who work in other groups. These are the earlier students who have contributed to this program, and these are my collaborators. I will stop here and take questions. Thank you.
Thank you. I think there is one question by Mr. Om Prakash. Are you there? If you can ask the question yourself.
Yes, sir, I am there.
Yeah, please.
Sir, good afternoon, sir. Sir, actually,
Yes,
sir,
please go ahead.
I want to know that, sir, is bacteriocin and antibodies working on same mechanism?
They have similar function. They are defending pathogens, but the mechanism is different. The information to make antibodies is in the host genome. So using the host genome, antibody is a protein which is being made, and that is interacting with it is defending with the virus or other pathogens. Whereas bacteriocins are chemical moieties. These are not protein. They are protein-like, some like peptides type things. They are small molecules, but the information to biosynthesize them is in the genome of the microbe or probiotic. The beneficial microbes have it. They make these molecules to fight against another microbe, which is a pathogen. So in the broader sense that they are defending us, you can say they are similar, but actual mechanistic details are very different. Antibody is a big protein, and bacteriocin is a small peptide with many cross links or things like that.
Okay, so Abhishek, I have some more questions regarding this. So in this case, can I ask the question?
Yes, sure. Please go ahead.
You are able to resolve the problem of PKS, I guess, yeah. And is this machine learning algorithm can also pick up the exact drug-like compounds from NRPS or terpenes? And also, we are facing a challenge that we are sequencing a lot of microbial genomes from marine resources and other resources such as lichens, and we are not able to tell exact conformation of the compound, or even not able to detect the compound structure. So how could we resolve such problems?
Okay, see, right now what we are predicting is the compound structure, chemical structure of the compound for these three classes of metabolites: polypeptide synthase, non-ribosomal peptide synthase, and RIPS. And again, since you seems to be you are familiar with the field, so within polypeptide synthase, this our approach will currently only cover class 1 modular. There are many other variants, or probably class 3 it can handle. But coming to your question, whether it is going to be a bioactive molecule or not, we are working on it, but we have still not applied machine learning there. What we are doing is we are just, once we predicted the structure, we are just comparing with known bioactive molecule. That is the example I showed. The molecule we predicted was similar to nisin. In that sense, you can say bioactive. But what we are working on to have a machine learning classifier which can predict bioactivity, again, that would depend on how much data we have and this one. And coming to your question of confirmation, we are currently not working on confirmation of these metabolites, but I believe this may not be difficult because these are likely to have very constrained structures because of a lot of cross links and cyclization patterns or things like that. But obviously, this is a general statement. Only when you look at the actual problem, you realize obviously there might be additional complexities there.
Yeah, so I have another question. How successfully you count from genomic side? When I'm coming, I see that NTS mass has massive amount of problems. So basically, it gives us very little information from genome to drug mining, but it's still one of the widely used software or application. What is your opinion about NTS mass and their machine learning algorithm? They are continuously working.
The anti-smash and all, see, this is all going to depend on what is the quality of the genomic sequences you have. So, and the problem comes like they are in gene clusters, and if you do not have a completely assembled genome, you have only contigs and all. If your cluster falls on the boundary of this one, then obviously your prediction is not going to be correct. But if it is in the middle of the contig and things like that, there you will be right. But this is what would decide the type of genomic data which is coming. But some of the cases, like using metagenomic sequences, even if you do not have complete cluster, you will be able to predict. But all these predictions are telling only what sort of chemical moieties are there. Even anti-smash or prism, which are the other two programs which have not handled the problem of cyclization prediction. And this our bioinformatics paper or earlier RipMiner were the other two programs, whereas what Prism does is it enumerates all these structures. Now if you have mass spec data, you can combine with it. So this is our first attempt to predict, and of course this is shown on a relatively small data set. As more people use it and we compare with more number of structures, we will be able to say what is the real accuracy of this prediction. But all these tools are constantly evolving, so that compared to this one, so I believe in future we will be in a better place. Like earlier, when many 20 years back when I started working on this, if you say I'm trying to predict metabolite, they will say, oh, are you sure? Can you really predict? You can't tell the substrate specificity even of a single catalytic domain. But that is what seems to be changing, because we have more data, we have better computational tools, and machine learning comes to help. So that is what the things are improving.
Yeah, I agree with you, and really, I'm really thankful that I'm talking to you, and I have seen you with a great respect in the secondary metabolite world, and it's my pleasure to have a discussion with you.
Oh, thank you. Thank you.
Sir, I have a question if you have time. I mean, this is Pritesh from Triple IT Allahabad.
Oh, sure.
Sure, namaskar. Sir, it's really a nice talk. I just want to ask you that how you have found out the accuracy part, because when you are saying that it's accurate, so what do you mean by accurate? How you found the structure is accurate or not? I mean, what is the accuracy function of scoring you have taken?
These are tested on the known data, because you divide the data into like a training set and validation set. How many structures you could correctly calculate the cross links. You know which are the cross links or how it is cyclizing. So if you predicted the same cyclization or cross link, taking into account the true positives, false negatives, and all these, the accuracy is calculated using that. But this is on the known data set. This is not on a unknown set. Then that on that you have to do a experiment, and that has to be compared, like what is done in the CAS prediction. So that is the actual blind prediction. But here, the known data is divided into, like any machine learning problem, it is estimated on that. Did I answer your question?
Actually, sir, yeah, I got your point. Like finding true positive and true negative, that's what you have told. But then my question is, how you know that this structure is the true structure? I mean, suppose you are having a structure X, and X has been predicted through your algorithm, and then you got a X predicted by your software. Now, how you are comparing that new predicted X to the already known X and saying it's the same structure?
I mean, how means you see the what is already known is a experimentally determined structure. Okay, so that information we have compiled from literature and kept it. Fine. So now the chemical structures are compared using what is called Tanimoto score. So you can compare with using Tanimoto score. If you are getting a Tanimoto score of 1, you are like your every
So,
atom.
that means, sir, you are actually checking the Tanimoto score of the predicted and the Tanimoto code of already known, and you are finding the edit distance. Is it?
Exactly. Tanimoto score is based on a jacket distance. Yes.
Okay, sir. Okay, thank you.
Okay.
Thank you, Debasis, for an excellent talk. I do have a few questions, but I'll ask them in the end.
Okay.
Let us move to the next talk by Dr. Abhishek Kumar. Dr. Abhishek is
Not
presently the
proactive.
head of
I
computational
think
genome biology.
proactive approach with a vision. And I
Can
believe
we
that a long-term vision with a short-term goal.
So before we
of
go
1 year
for
is
the
important.
next talk, I would thank
And,
Professor Devasis Manti sir for his
I'm
excellent
very afraid that
talk,
I will not be able to
and
spend a
we are
lot of
really
my
grateful
time.
that he gave his
But I have a
time
team of at least 5, 6
really
scientists
enlightened
working
us with
with me.
his
And
interesting
they
work.
will
Sir, thank you,
be
sir, very
like
much.
Nagamani, Hriday Jyoti.
Okay, thank you, Devendra. We'll move on to the next talk by Dr. Abhishek. Dr. Abhishek is presently the head of Computational Genome Biology Laboratory at Institute of Bioinformatics, Bangalore. He had earlier worked in the University of Kiel and German Cancer Research Center in Germany. He also previously worked on understanding of next generation sequencing data, implementing a pipeline for the NGS, and to annotate some non-referenced eukaryotic genomes, such as invasive species Colorado potato beetle in biology department of University of Padova, Italy. He obtained his PhD in bioinformatics from the University of Bielefeld, Germany, and an MSc from Madurai Kamraj University, India. And he is a recipient of prestigious Ramalinga Swamy Fellowship Award by Department of Biotechnology. And he is employing multi-omics approach for studying cancer and neurological disorders and developing computational pipelines for detailed analysis of genomes. He has more than 60 published research articles with an H-index of 17. May I now request Abhishek to present his work. Dr.
Yes,
Abhishek.
can you hear me? Thank you. Thank you very much for a nice introduction. Just confirm me if you can hear me. Let me see.
Yes,
If
we are able to.
yeah, okay. Good. So thanks everyone in Bioclues and Dave and Prash and everyone to give me an opportunity to um, uh, give a presentation in this INBIX 2020, and I'm going to tell you about what we did with Hodgkin's lymphoma families and their genomic study. And let me briefly tell you at the start what computational genome biology in a laboratory which we have set up recently in Bangalore at Institute of Bioinformatics is doing. So my laboratory is interested in biodiversity genomics and with different areas of genomics, and also we are also interested in the biodiversity in terms of genetical as well as biochemical. So previous talk when Professor Mohanty has already introduced you to natural product discovery. This is one of the areas on fungal and lichen and marine fungi we have extensively worked, and recently our students have done a great job on several competitive lichens, and the current in the we are also working on different human diseases, and today I will discuss one of them. And then at the same time we have different areas where we are working, and currently we have funded by two Department of Biotechnology project. One is disease genomics platform development, and one is about genomics of Asian green mussel, and one with ICMR, with population genomics of Wucheria, with help of an ICMR team. And with this, we'll go and introduce you why and what we are doing. So as when I finished my MSc out in 2001, human genome came, and you know that it costed massive amount of dollars. And that point of time, people were very carefully asking what I want to sequence, because that will cost me billion of dollars. And depending on size, definitely, but human genome was is a hallmark of the genomics. At that point it was very expensive, but parallelly people were working on short read sequencing methods, and that has changed the whole dynamic. So today you can actually sequence entire genome in less than $1,000, and if you are working alongside a genomic center or a center core facility of genomics, you can even bring down using Illumina HiSeq to $500 or so. So that means in 70,000 rupees, I will have my genome in my hand. And that puts that reverse challenge actually. People were asking earlier what we want to sequence. Now you can sequence everything, and then now you have to ask why you want to sequence. And then we have to also ask, do I need to sequence, because maybe somebody else has sequenced already something. If you want to resolve a problem, there's a lot of data already existing, and that led into another problem that Moore's law is saying. In the bottom I have shown that everything is improving in a linear order. Suppose iPhone 1 to 12, there's no massive change. They go with a linear pace, but here the entire pace has shuffled, and that's what this is not following the linear line, but it's rapidly decreasing. Those who are coming from different areas will be able to understand. So with this will lead into a massive amount of data which is already existing, and data is doubling every 6 to 7 months. And with as you are aware, DBT has already 1,000 genome sequencing where Pritish is participating, but then there are several other data sets, and every now and then people are reporting that they want to cover entire planet, entire uh, uh, entire plant species, insect species, and that massive amount of data is coming, and that data will exponentially grow in the over a period. So where we are working on the surface of the data, and we're trying to analyze the existing data, and while doing so, if we have some sequencing project or where we have partner who have sequenced the data, want to make sense. So basically we are data analysts, and we are analyzing data. My team is focusing on data analysis, and many of my team members will be presenting on what they have been doing on different area of the work. But we are focusing on how we will be ready for a new challenge in data analysis. And do we have the computing power? Do we have the human skills? And so we are growing those skills. And while doing so, we have developed several projects. But one of the projects which is very interesting, Hodgkin's lymphoma, I'm going to discuss. So some of you might not be coming from genomics, so I will tell you what is genome sequencing. Suppose you have a human genome. For human you want to sequence, you first time do it is called genome sequencing. But once we have human genome available, you want to seek. If I want to sequence today my genome or a population genome, is called resequencing project, because reference is already present. So you have a prior knowledge of how genome will look like, what will be this size, and then you will do the population genomics for population genetic purposes. For instance, seeing how the population of Karnataka is different from the population of Bihar or something like that. And similarly, you can study a healthy individual versus a person who has cancer, or say even you can use it in other diseases such as cardiovascular, rare diseases. And so this the second line of after first sequencing in any species, second line of sequencing is resequencing, because the pre-existing data, and from there you will try to see which are the mutations from reference with respect to population or with respect to disease is actually what are the changes that leads into a point mutation or a small or large fragment deletion could lead into disease or change in the population dynamics. And so this is what since we are doing human as a reference, we are calling human genomics, but if it is a fish, it will be a fish genomics using a detection of the point mutations which will lead into changes. And so the data sets which are primarily used in population is 1,000 genomes, a genome ad, and once the Indian population database is available, it will be also added here. UK data set is already there. And then in cancer data set, we have Cosmic, CBioPortal, ICGC. And if you are aware, if you are working in the area here, you are already aware that in India is participating with oral cancer in ICGC. And so what is the main trend? So you try to find out the mutation and imbibe into the protein, or you find a non-coding mutation, imbibe onto regulatory elements of the genomics. Regulatory elements, it's say enhancer promoters or super enhancers or certain other areas of say non-coding RNA. And so it can be a regulatory effect, or it can be an effect on the protein sequence function, structure function. So while we were doing that, we developed a method called familial cancer genomics pipeline. And so in what our idea was that you have cancer in families. So some members are healthy, the red ones are non-healthy, and the white ones are healthy. The circle is female and the square is male. And so some of them have cancer and some of them are healthy. Why is it happening? And so if you want to know, if you know this, that you can actually build a better prevention methods. If I can tell my child, a boy, I have your genetic map, you may be getting these diseases because me and my father has it, so on. You can build up for any disease. And so Time Magazine has cover want to know my future, and so on a similar concept, what can a kid can get in future. So can we find the markers from these families to imbibe in genetic counseling? So initially we developed a method for only using whole exome sequencing. Whole exome is only coding region, which is 48 MB of human genome. And that the strategy was successful. And then 48 MB out of 3,000 MB is very few numbers. So if you're realistically understanding non-coding genome, will have a major fraction of genome. And so then we started doing whole genome sequencing, and we improvised our pipeline from version 1 to version 2, and then both of these versions are published. And then we are still thinking, even if we do whole exome sequencing or whole genome sequencing, we are still left out with some families where we cannot deduce a final mutation or final genes or pathway genes where mutation is. Can we do a pathway stratification based method and develop on the next version or improvise the first two version? And that's when we are thinking about it, and we are working on it. And so this with this method already existing, I will go briefly how we are doing. You take your take your genome sequencing, or either you take whole genome sequencing or whole exome sequencing from a family, and do mapping, call variants. I'm going rapidly so you can read the paper. and then you can call the variance. And once you have variance, you can annotate the variance. What annotation means in the variant annotation means already known public resources. I told you, you match your variant to those public resources and see whether your variance is known in public resources or unknown in public resources. So it's like doing a blast of if you're aware of blast, doing blast of 0.1 nucleotide at a time, and matching. So you can match positions. Yeah. So and then once you have that, you can always filter the variant for the good quality. What is good quality means? Good quality means a coverage. It should not have less than 5 read coverage anytime in the sequencing, quality of 20. And in our case, it was a family. So in family, that understand when you are if disease is prevalent in a family, that means it's not prevalent in the population. So it has to be lower frequency mutation, and therefore minor allele frequency has to be very low, and that is decided on based on the population size and all. See, these samples are coming from European population. It's a 0.1% of 0.1% of MAP was enough, but in India, since population is very massive, we can even lower it down. And once we have those variants, we pass pedigree segregation means we will allow these cases to have the mutation and these controls will not have. Sometimes we'll allow some cases depending on the pedigree. So basically, we'll discuss with doctors what could be how to deal with it, and also genetic experts. And then we pull it out, and then what we will do that after that we'll do the first round of variant ranking using a machine learning based tool called CAD, and then it will give you a ranking, and then divide the ranking in the coding region, 3', 5' UTR, non-coding region. CAD is not very powerful in dealing with non-coding variant. Probably you are aware that no tool is perfect at the moment. So we will take that score, plus we'll use without CAD another strategy. We'll explain in next slide. And then we'll look deleterious mutations in coding region by using SIFT polyphen like 12 tools, and try to make a consensus. And then we also cover intolerance score. Intolerance score is something like that. If you have a protein which has 1000 mutation, 1001 mutation will not be a big difference. But if a protein has only 5 mutations, 6 mutation will be critical, because the protein gene is prone to have less mutation. So those scores were invited by 5 tools, and then we look what are the region for 5' UTR here. If there is some binding happening in 3' UTR, or a micro RNA will bind in 3' UTR or 5' UTR. There is a regulatory elements, and these will try to find out, and non-coding region will combine similar method. And then we will invite these mutations on association plot and regulatory plot using genome browsers, and association plot using a special kind of browser which only looks for the variant. It is a genomic map only, but focusing on association and linkage disequilibrium. And then once you have that, you will try to mine with other resources, like are these mutations are these variants are mapped to cancer predisposing genes. CleanVar has, due to this mutation, is there RNA or protein expression change already known in the public data sets? And also, if there is a phenotypic changes or chromatin changes, are these mutations are germline mutations, somatic mutations? And also, you have to recheck before we give the final result to a doctor. We will recheck whatever computationally we have done. We recheck the sequencing output by running sequencing browser called IGV. You can have a look. It's open source. So basically, you can see your reads mapped to, and the region is good or not. Because suppose your entire pipeline had made an error, you reconfirm it if there is something because of repetitive elements, transposable elements, some sort of error is happening or not. If it is perfect, you always, that is also point to report it. And in non-coding genome, what we had done, that we had taken super enhancer and promoters, and then uh, try to look if the mutation is within the same range. So basically, you have the position of mutation, you have positions of the enhancer and super enhancer. You just use the BET tool to find whether this is matching within the enhancer or promoter region or not. And once that's done, you can check whether this is transcription factor binding site, there is a chromatin chain, there is a genomic element segmental changes used for segment. These ultra conserved regions, uh, ultra conserved regions are regions which are conserved from human to chicken fully. And then ultra sensitive region from FUNSIC, which is a non-coding region, which is very sensitive if there is a mutation that will lead into change in gene expression. And then combine this, look back again, expression and motif change, and go back to the initial pipeline of regulatory nature. Okay, so once we had this pipeline was generated for the normal purpose, and we had analyzed different cancer families, say about 100 families, 400 members, and about 50 to 60 new cancer mutations. And then in Hodgkin's lymphoma is one of the case we applied. And some of you might want to know what is lymphoma. Lymphoma is the most common blood cancer, which is a white blood cancer, cancer of white blood cancer in lymph node. And these are the symptoms. My cancer is a very dangerous disease. So whenever I'm discussing science, I'm also discussing symptoms. So some of you want to know it. So it's a swelling of lymph nodes. You get fever, very night sweats, tired. You will be tired, excessive weight loss, no appetite for food, and body itching, abnormal pain and swelling, coughing. So basically, you can go and look at this website given here, cancercenter.com. From there, I have taken this slide, and one need to, if you have a question later on, it's better to answer in the talk itself. But then what we as I told you, the lymph nodes is shown here in green, and then the white blood cells based cancer of lymph node is lymphoma, and this is how the strategy is. As I shown you before, you look into genic and intragenic regions and try to see the mutation. What people were doing, initially to find high penetration or genetic architecture linkage analysis will be performed. And for say from 1960 onwards, and the cancer genes are initially detected using linkage analysis. And the second way is to look at the common mutation, common low penetration gene by GWAS, and then some analysis were performed by gene-based sequencing, and people found some new cancer genes. And now, with help of whole genome sequencing, we started whole family sequencing of whole genome sequences of high risk cancer family, and that will lead into rapidly find mutations which are laying in moderate to high risk region. So whole idea is that you take cases with a strong history, and then you have some unaffected controls in the family, and then you have also older affected persons, and combining the family history, can you find a single gene mutation that could be cause of a disease. With that idea, Hodgkin's lymphoma is a very high familial risk of 5.9 was already found by in the same team. Frank has already reported in 2017, and then there are a few high penetration genes were already known by other groups like KDR, POT1, NPAT. And then genome association study had shown that there is a HLA based association, and also 18 risk loci were identified, which were germinal center dysfunction, T cell differentiation, and NF-kappa uh, activation were already known. And there are other risk factors like viral infection. And so while we were working, we found two mutations, and those mutations are DICER1 mutation, and also we have pull out whole data in whole pathway context and identify the pathways. So I will go and explain you. So these species are three families. We have two cases in first families. In second family, we have uh, total 5 cases, 2 are dead. So basically 3 cases. And then we have third family with 2 cases. So when we combine those cases, when we did the whole genome sequencing, we carried out a combined analysis. 3 families initially variants were numbered in lakhs or thousands, and then we did a pedigree segregation. We get lesser number of variants, and once we have done the mapping on the exomic 3' UTR, 5' UTR, the number even go down. And when you combine with the CAD score, the final 5' UTR variants were 46, 3' UTR variants were 56, and then 23 were exonic variants. And on top of it, if you look at the cancer predisposing genes, we've in 3 families there are 9 of them, and 48 were there's a data set of causative HL genes. And then we identify these in each family. We identify some genes which are predominant or prioritized by our pipeline. So these are genes listed here, and DICER1 we have carried forward. And so these are the mutations. If you look carefully, so there are more mutation. There are 10 to 15 mutations in the family 1, and then family 2 has few mutations, and family 3 has more mutations. And then you have also some of them are uh, frame shift and stop gain mutations. So we have listed them, and when we map those high rank mutations on the pathway context, and the orange ones are the genes which are affected. So what we see is that we found that cancer and hematological disorder, immunological disorder network was enriched. In the second case, developmental disorder pathways were enriched, and third, the key case, we have post RNA translational modification, cell death and survival pathway was enriched. So the orange ones are the genes where the mutations are mapped. And in this study, we also in a larger cohort with 3 families, we identified mutations in DICER1, and independent study also we have identified DICER as a candidate mutation, and this can be confirmed by Sanger. And again, when we looked into the mutation, where is the mutation is located? Mutation is located at C terminal end at exon 24. And when we check the mutation expression profile of known tumor suppressor genes, we see that there is a regulatory effect, and this was then also controlled where checked for the negative regulator of let 7 family members, LIN28 and LIN24. So combining these, what it says that what we found that our pipeline is capable of picking up coding variants and also regulatory variants. And in these cases of 3 HL families, we have identified 49 coding variants and 79 non-coding variants. Plus, we have identified the map of genes were mapped to the pathways related to the B cell activation and proliferation and cancer, hematological disorder, immunological disorder. And this is this similar findings were there from others group as well. And in one family we have one missense mutation in DICER, and this is impaired. This impaired the expression of tumor suppressing microRNA, and therefore it is this is potentially used in HL biogenesis in this family. And with this, I would like to thank our big team at German Cancer Research Center. And this is the main building here. And these are the funding sources. And I think we have a picture here. Yeah, so this is the TP3 building of German Cancer Research Center, where the entire top team, I think most of the people in the German Cancer Research Center were sitting in the left side of the building. And our team used to sit on the third floor. And still the members are sitting there on the third floor. Okay, so with this, I will end up, and now I would like to introduce you my new team. And these are my team, and three members of my team: Gaurang, Vineet, and Pradeep will be presenting their work in the coming session. And we'd like to invite a new trainee or new people who want to join our lab. Can discuss with us via email, call, whatever way you like, and you can visit our website, see what we are doing. With this, we'd like to thank everyone again. Hope I'm on time.
Thank you, Abhishek. It's a very nice presentation on this thing. If we have any questions, can we take one or two, or otherwise we'll move on to the next presentation. If there are no questions, or you can ask in the end also, because we all will be here. We'll move on to the next presentation by Dr. Prashant Kumar. Let me introduce Dr. Prashant. Dr. Prashant has received his PhD in molecular biology in 2009 from the Max Planck Institute for the Infection Biology in Berlin, Germany. Following this, he undertook postdoctoral training in the laboratory of Professor Carl Simin at the Department of Cancer Biology at the University of Massachusetts Medical School in USA, where he established a novel mouse model of malignant breast cancers. Subsequently, he joined the laboratory of Professor Jean-Paul Thierry, a world-renowned authority in the area of epithelial mesenchymal transition at the Institute of Molecular Biology and Cell Biology in Singapore. During this period, he was involved in the development of a novel point-of-care diagnostic platform for detection of bladder cancer biomarkers. He is the recipient of prestigious Ramanujam Fellowship awarded by Department of Science and Technology, Government of India. At IOB, he is focusing on using the genomic and proteomic approaches to study cancers. He has more than 45 nationally, internationally published research articles. May I now request Dr. Prashant to present his talk. Dr. Prashant.
Hello, can you hear me?
Yeah, we can hear you.
Okay. Can you see my slides? Can you see my slides?
Yes, we can.
Okay. So thank you very much. I think this is extraordinary time, I would say, because you all know that the world is going through the crisis time. And what I have realized that even though the COVID has taken and brought many of us in a very hard time, but the kind of meetings which has been held last 6 to 8 months, you would see that we are pouring a lot of information, and this is giving opportunity to the students or the colleagues to share their work, to learn about each other work horizon, and this is where I think this is a big achievement over the time, that even though it's a hard time, but the scientifically you would see that we are doing good, and this is how I think it should be taken. So I think it's a great work by the committee who has arranged this meeting, because I would again repeat that due to so much limitations you managed to do this, and I could see that there are good number of students participated in this meeting. So I would try to quickly talk a little bit about the basics of NGS, because I think it's very important to understand that what is something NGS is doing. And the title, you could see that I'm mostly going to talk about the translation part. that what is something you could translate, because no matter what technology we are using, but how you could use this technology to answer some of the queries which is really baffling us. So NGS in targeting cancer therapeutics. So it will be therapy, that how you would identify a new therapy which would make sense for the patients. So very basics, if you talk and if you understand that how the molecular machinery works. So there's any mutation at the level of a genome, at the level of gene, and this is why you identify the mutation. So at the DNA level, at the gene level, there's a mutation. It would also encounter any epigenetic modifications. And further, when a DNA, so it's a gene which is getting transcribed, it makes the mRNA. So this is where the another level of modifications come, which is called post-translation transcriptional modifications. And this is all these three different kind of modifications, we keep it in a box, which we call it genomics. So whenever we talk about the genomics, it's not only necessarily we are talking about the modifications at the level of DNA. It is also at the level of RNA, even post-transcriptional. So then what happens, and this is the center dogma which many of you know, that how the gene transcribed to mRNA and mRNA makes the protein, and this is where we call translation. And translation, when the protein forms, there's another level of modifications, and which we call it post-translational modification. And this is again different kind of modification at the level of glycosylation, phosphorylation, acetylation. And the new area which we started working on is metabolites. So now the world has moved forward beyond genomics and proteomics. Now we are talking about the small cell molecules, which is secreted by the cells and which is in the vicinity. So this is where we call it metabolomics. So what would you realize that when you move from genome to proteome or metabolome, what is also happening is we are moving towards the phenotype. And it's a cellular phenotype. So when we talk about the genome, it's a mutations at the level of gene. But when we talk about the protein, so you are talking about a modification which actually express or reflects in the phenotype. It could be cellular phenotype, it could be overall organ phenotype. So very briefly, quickly, I would like to touch base that when we talk about the NGS, it has different level of work flow, which starts at the isolation of DNA, library preparation, NGS platform. You use different kind of NGS platform. Then you talk about the raw data, and the raw data comes when you align. And when you align, then you ask that what is something you are interested. You are interested to understand the expression of the gene. This is where we call RD-seq. So it's called transcriptomics. So or it could be DNA-seq. So and further, what happens that you would start doing the downstream analysis. So in a nutshell, what you see, any genetic variation, it could be at the level of one particular point mutation. So a single nucleotide changes. It could be microsatellite where the changes are bigger, and the insertion, deletions, focal amplifications, translocation, and nucleidi. So these are the different kind of variations. When we talk about the genomics, it includes such different kind of variations, and all depending upon the length of the bases, how big the changes happening in the genome. And it's a very good example. You could see that a single nucleotide changes, how you identify. And there is a reference, and the normal, you can see that the normal sequences look like a base coming one after other. But what happens in a diseased condition, a single base is changing. So C become A, and this is where what we call it, the tumor, you are identify that single SNV, single nucleotide variations. So I would say that this is amazing that the way we have sequenced once we established the NGS platform, and this is only 2014, but now 2020. So they are in number. So what I'm mentioning you that fungal genome, microbial, plants, and many more. So we have already sequenced, and you can see the era. This era has changed enormously after we establishing the NGS platform. So the cost, this is something very challenging. You can imagine last 20 years, the cost has reduced enormously. So 2001, you can see the cost, and over the time, the cost has decreased enormous. And now you talk that, okay, you pay $1,000 and you could do your sequencing. So you want to sequence your whole genome, You pay 40,000, 50,000 rupees, and you can sequence your genome. So this is something amazing, and I would say that over the time we will improve, and the price will reduce a lot. So I would talk that sequencing based test, when we talk about whole genome sequencing. When we call whole genome, it includes all the genes. Whole exome, where we are talking only about the exonic region, and then there are different kind of panels, large gene panels and small gene panels. Then we go forward where we talk about the hotspot panel, and this is very much used clinically. When the clinicians are very interested to find that, okay, what are the hotspot mutations present? And then the transcriptome and targeted RNA panel. I'm skipping it because 15-minute time, and I wanted to give some background before I talk something on the cancer. So see, this is where the TCGA, you have heard that it's amazing platform, and you actually reservoir your data, and you see the TCGA. Now you could see that these many numbers of data is present. So what we call it 1 petabyte, and you could see that how many DVDs would require to actually acquire these a huge amount of data. So what have been done, there are 30 different tumor types, and I again telling you the numbers, these are very dynamic number, the number keep changing, and there are these many patient and different kind of data types are already deposited in TCGA. So now if you talk about the genomic alteration or genetic alterations, and this is such a beautiful example in case of colorectal cancer. You could see that a mutation could clearly change the normal colonic epithelium to adenoma, and the adenoma progress, and what is the phenotype, you could see the carcinoma, and this is the effect of a mutation. So this is something very extraordinary. I would say that the work which has been published in Nature, and what they have done, that they have actually accountable for the somatic mutation load across the different cancer types. And what you would realize that the top cancer where the mutation load is much higher is melanoma, and that makes sense, followed by lung cancer, bladder cancer, esophagus cancer. So this is where we were very interested that the bladder cancer, which I was not at all expected that the tumor mutation burden will be so high in bladder cancer. It is ranking almost second, melanoma and lung. And again, you could see that the way you talk about the different kind of alteration in bladder cancer. So one of thing is that you could see there are very recurrent mutation, and P53, FGFR3, RB1. So many of the tumor suppressor genes getting mutated. And what we know that there are two fusions in bladder cancer, FGFR3 and TSCC3. So this is something you found at least 2 to 4% of patients present with this particular mutation. And there's another fusion with FGFR3 and BAIA P2L1. So these are two fusion product actually, which has been very prevalent in bladder cancer. So this is something very interesting we have done. That way, so see, most of the cancer, when we talk, we call it's an old age disease, and what happens over the time, the mutations, the aberration happens, and that accumulate. But very interestingly, this particular case where a very young guy got the bladder cancer, and we call it the bladder cancer median age is 72 years. So for you to get a bladder cancer in an early stage, what is something happening? And this is where the bladder cancer patient who had been presented, and this is what PGI Chandigarh, this is a hospital in Chandigarh where this case had been presented. So you could see that the tumor progressed, and the patient presented with the gross hematuria, and you could see that on day 72 the patient demise. And what you would see, this is immunohistochemistry. What you could see that the arrow, it shows that the tumor is actually invading in the muscle. So it's become the muscle invasive bladder cancer. What we have done, we were very interested to do the sequencing. So we have done whole genome sequencing and somatic mutation. You could see the numbers of SNVs, missense mutation, frameshift, nonsense mutations. So you could see that there are quite a high number of mutations. And we have done the interaction with the identified gene harboring somatic mutation. And what interestingly, we found that there is the enrichment of ion channel pathway. So many of the molecules in the ion channel pathway were mutated. And this is what you could connect and connect the landscape of somatic mutation in bladder cancer, where what has been known earlier that there's a mutation at the level of NSD gene or CTNND1, and I told you about the P53 and FGFR3. And these are involved, their mutation leads to the DNA repair pathway and apoptosis senescence. So what we identified that when there is the different ion channel pathway getting mutated, what it leads actually to more cellular proliferation, more tumor growth, more cell migration. And this is something very, you would say that very unique, because no one has reported that the ion channel pathway has mutated in such cases. And this is what you could see, that because when you are performing such kind of NGS, your expectation is that either you would identify a novel mutation or what is something which is existing. And if you see that a clinically significant mutated genes, you could see a potential drug. There are already some of the drugs available for P53, RB1, or some other. So like sodium ion is there, potassium ion. So there are some of the molecules which have been identified already, and there are potential drug available. So what actually it pushed you towards that, if you identify some of this novel mutation, you could definitely identify that if there is a drug, preclinical or clinical drug available, even it is at the level of clinical trial phase 2, phase 3, it could be given to the patient. And this is something remarkable. You could see that some of these drugs are already in the clinical trial for some other cancer like breast cancer, cervical cancer. So I would quickly switch to another the unique study we have done, and this is with TMS, Tata Memorial Hospital, Mumbai. So this is done on the metronomic chemotherapy. So I don't know how many of you know the metronomic. So metronomic is something where you give the patient the low dose of chemotherapy, and it would be for the long period of time, because you know that the many of the time what clinician realize that the chemotherapy has an adverse effect and the patient may not able to tolerate. So now it is the another paradigm where the patient has been given the metronomic chemotherapy. So what we were interested that what happens over the time, and most of the chemotherapy, you know that the patient stopped responding, and this is where we call the non-responder group. So you could see that what we got 6 patients, and 6 patient has been selected for this metronomic therapy, and after 1 month and after 5 months, we have again taken the tumor tissue and whole exome sequencing was done, and you could see the 2 group, responders, non-responders, and we could only got the result about the responders, because the non-responders, they didn't survive. So these are the different cohort where we have done the whole exome sequencing. So what you would see that this is showing the somatic alterations across all these responders and non-responders. And these you could see to the down that there are 3 different time point. I told you that pre-treatment, treatment post 1 month, and treatment after 6 months. So what you would eventually see that we got a good number of synonymous and non-synonymous mutation. And of course, you are more interested about the non-synonymous mutation, and you could see that mutation per MB, it is showing almost more than 8, and which is something which has been reported in head and neck cancer, that the mutation per MB is more than 8 or 9. And what we identify, there are 2 actually mutations of the Notch1 and SYN1, which we identified in case of responders compared to non-responders. So this is where the somatic mutation types you could see that we found between pre-treatment cycle 1 and cycle 2. And this is something, the signature we identified in responders and non-responder cases. So what it actually eventually gave you that there are some signature, there are some mutations. However, we didn't find something very exclusive. And the reason being is that every sequencing, when you are performing, you may not expect that everywhere you'll get a very unique signature, because the patient has been given the treatment post 1 month, and post 6 months you are collecting the sample. So you may not expect that there will be huge number of somatic mutation or something, a very unique signature you may identify. So this is again a finding. So quickly, I would talk about the liquid biopsy, because I think many of you have heard recently about liquid biopsy. And in a very simple way, what is liquid biopsy? Liquid biopsy is something that you just get the cell from the blood or DNA from the blood. Now you have 2 different layers from the blood. One is plasma, and the other where you have the blood cells, which is sedimented. So the plasma, where you can identify the DNA, and if you can identify the DNA, it would be used for further knowing what are the different alterations at the level of DNA. And this could be amplification, translocation, point mutation. So all such kind of different alteration at the level of DNA. And this is the beauty that the liquid biopsy, when we call, this is minimal invasive because you may not need to go surgery multiple times. You just get the blood sample from the patient and you ask the similar kind of questions that what kind of alterations present at the level of DNA. So this is a study which has been published in Nature Medicine, and this is a work from Caroline from UK. So what they actually tried to understand that if this is a small cell lung cancer patient and they wanted to identify the signature between chemorefractory and chemosensitive, if you could identify a signature doing the whole genomics, and that signature could be used to identify if the patient will respond to the therapy or patient may not respond to the therapy. And you could see the numbers were n is equal to 6 and n is equal to 7, 90 days, 3 months. So most of the time in the clinics, when you make a call that patient is responding or non-responding to the therapy, we give the time window of 3 months. So what you would see that what they identified, there are 16 CNA copy number abbreviation. So they found 16 different signatures, and they just arranged it with the profile 1, 2, 3 till 16. And of course, you could see that with the sensitive and refractory patient, you could see some of them refractory where there is gain of copy number and there's loss of copy number. So these are the signatures they identified. And this is how it looks like. If you cluster these signatures, you could see that there is a beautiful unique signature between chemosensitivity and chemorefractory. Down, you could see many of these genes showing differently with the chemorefractory. So what they have done, they have already the training set which was ready, and then they have tested. So you could see the training set, the accuracy was 100%. So you took 13 patients and the training set has been established at all 13. Based on that, they made a chemosensitive, so sensitivity and refractory. Now they have having the testing set. They have done again copy number analysis, and out of that you could see that again out of 18 samples, they were succeed using the similar kind of signature on 15 patients. So the accuracy of this kind of testing is almost more than 80%. And this is something extraordinary that you could use. And again, this number could vary again if you go and such kind of experiments could be done on the largest cohort. The goal is that how you're utilizing the information based on the genomics. And this information using the liquid biopsy, you could eventually use to identify such signature and could be used to predict if the patient is going to refractory or going to be sensitive for the chemotherapy. So this is something which is, we talk about the cell-free DNA. So this is another study from 32 lung cancer patient during each chemotherapy cycle. Now you can again see the beauty of this liquid biopsy, that you can do the longitudinal access of the sample and you could predict how the cancer behaved. So the blood sample has been taken, DNA has been extracted, pre-chemotherapy, and then the patient has been on the chemotherapy, post-cycle 1, post-cycle 2, and cycle 3. So this is where you could see that again they found different kind of alteration between 27 pre-chemotherapy sample, and they even compare with the TCGA. So you could see that there is the alterations, and this is where they have compared with the study from the TCGA. So the distribution of all detected SNVs you could see in pre-cycle, cycle 1, cycle 2, cycle 3. So you could identify there are some non-silent mutations where there are some substitution from C to G where there, and you could again go and realize that every cycle there are the changes, there are some changes happening at the level of SNVs, and there are different kind of mutations which are accumulating. So this is another unique example. You could see that how liquid biopsy was used actually for the therapeutics purpose. This is the patient, it's a lung cancer adenocarcinoma patient which has been presented, and what you could see, it is in the year 2013. And most of the lung cancer patient which has a mutation of EGFR, and for EGFR, this is a panacea that you give the gefitinib. So this is the targeted therapy, and you could see initial diagnosis EGFR mutation, gefitinib has been given. Gefitinib after 2 cycles, 2014, the patient has actually accumulated the new mutation. And this is again the EGFR T719M. So this is again a very well established mutation. The patient become resistant to the first line of therapy, and there is another line of therapy which is osimertinib. So osimertinib has been given, and what has happened? Then you could see that there's patient again relapsed, and there's a liver metastasis. You could see this scan. So now at this point of time, it was very difficult for clinicians to ask the tumor tissue. So what they decided that why not they use the liquid biopsy, because the patient was not in a great condition. So you cannot ask patient to go for surgery and again to evaluate at multiple time point that okay, you could collect the tumor and do the NGS. So what they did, they collected the blood, and they collected the ctDNA, and again they have done the NGS, and what they identified that there is a MET amplification, and you could see there's a MET amplification of more than 10 copy number, and this is where they realized that there is a therapy for the MET, and they have given osimertinib together with the combination therapy, so crizotinib, and you could see that the patient responded very well, and they have collected the data till almost in the year 2018 end, and the scan has been improved enormously. So this is something very interesting. You could see how you could use the information using the NGS platform, that the liquid biopsy could be used not only for the diagnosis but for the therapeutic purpose. So this is all long debate people do in the area of liquid biopsy, that what could be the best way you can do such kind of analysis. Either you would use predict, or you will use the CTCs, or you will use the ctDNA. And this is something has been done. They have done the copy number changes, alteration. They have checked using both CTCs and ctDNA. And what you'd see that more or less it shows similar kind of pattern. Either it is gain or loss. And what you could see here, what is in the green, it is showing the gain, and the red is the loss. And when they compared with the similar set of sample from the similar kind of patient, they identified similar kind of pattern. This is another study where they again wanted to understand that what would be superior to use, because sometimes it's very challenging to get the circulating tumor cells. So again, this was done on multiple myeloma sample, and you could see that the 3 samples they have checked, and again the alteration shows similar kind of pattern. It doesn't matter you use CTCs or ctDNA. So the complementary, so there would be another way, if you could access both ctDNA and ctDNA, and you could do similar kind of sequencing. Do you think that it will accumulate the information which we may miss either using CTC or ctDNA? And this is where they have done, and you could see that doesn't matter combining the data from CTCs and ctDNA, you may get hardly some benefit, but more or less you would have, or you would be good using either of the DNA for all downstream analysis. So the future, what is something we talk about, the clinical application of next generation sequencing. So the patients, you could see the patient cohort. And this is the cohort where we have not segregated that this patient cohort is responding or non-responding. But if you take the information based on the genotyping, it could be at the level of SNVs, it could be at the level of CNVs or gene fusion. Once you have the information, you could go next stage. And this is where you could identify those cohort who are going to respond to the therapy. And this is where we are dreaming about the personalized therapy, that the therapy is so personalized that you have information about the every individual genome, and you can target the drug based on the alteration on the patient. And this is what it shows, that you have done the personalized therapy, and it is targeting a drug by A or drug targeting by drug B. And the impact is that you could monitor the disease progression, and you could, based on that, again you make a call, and you can identify a new mutation, and you can again retarget with the drug number C or drug number D. And this is where you can also identify the resistance mechanism and re-challenge patient. So I would conclude my talk here, and I would just again bring the focus that it's a huge contribution, the way you have seen the scientists across globe utilizing different kind of technology, either it is at the level of proteomics or genomics. But the goal of the researcher is very clear, that they want to bring it to personalization, where it could be used, these kind of platforms, to answer many of the questions and to use it in further to understand the disease, how the disease evolution happens, and how we can use the information to find a better therapeutics. Thank you very much for your attention.
Thank you, Dr. Prashant. We can take one or two questions. Prashant is raising his hand.
Yeah. Thank you so much, sir, for allowing me. It's an excellent talk, Prashant, and also a wonderful talk by Abhi earlier. I'm just a bit curious, getting these non-invasive biomarkers, taking in the CTD and your CFDNA is certainly, a kind of a complex and yet a kind of a challenging thing. I mean, how do we see this particular thing, at least in terms of, the endometriosis, that many women, would undergo in India? Do we see this as a kind of a looking into the realm of the future coming up, Prashant? Thank you so much.
Yeah, so that's a great question, and I'm very happy that you are bringing this. So, yeah, hello. Yeah. So, see, it has actually changed. If you see the way the last 1 decade, we were talking long back about the NGS, not the use of genomics. And now you suddenly see that everywhere the clinicians are talking about the liquid biopsy. And you'll be surprised to know that many clinics in India, forget abroad, in India, they are routinely using liquid biopsy. You go to Kidwai Cancer Hospital in Bangalore, or you go to Mumbai TMH, when the patient come with the lung cancer, and of course, if you know that in Indian prospect that the EGFR mutation is very prevalent. So there's a very targeted sequencing. They ask you that simply you just give the blood, and they isolate the ctDNA, and they just do targeted sequencing for EGFR. And based on EGFR, they found mutation, they just give gefitinib or ilotinib. So this is BCR-ABL, amazing example. That this is one of the best example, BCR-ABL, where you give amitinib, and this works very good. So the way it is working, that yes, it's not an easy task, especially for circulating tumor cells, because we are working on CTC. I didn't present about CTC because I work on liquid biopsy, but I was just giving talk on the genomics because the science is, it's converging. You need to work on everything which is coming on your way. So what has happened that CTCs, it's not easy because these are rare cell population. You need to get these cells from the blood. You need to do the enrichment of the cells because you need to reach the number, the DNA, the quality of DNA and quality of RNA. But of course, the ctDNA way, it is very well used by the clinician, FDA approved. Now, many of these tests are FDA approved. So people are using very routinely in the clinic to identify the mutation, and based on the mutation call, they plan the therapeutics.
Wonderful. Thank you. Yeah. Thank you, Ken, sir. Thank you, Prashant.
So we will now move to the oral presentations. May I now request Dr. Amrish Kumar, who would be speaking on workflows for genomic variant discovery.
Yes, sir. I'm ready. I'm just sharing my screen and uploading my PPTs.
Yeah. In the meantime, you introduce yourself. I mean, I have.
Yes, sir.
Yeah, please.
Yeah. Is my presentation visible to everyone? Hello.
Yeah.
Yes.
We can do the presentation.
Is my PPT visible?
Yeah, it is.
Yeah. Thank you so much, sir.
Yeah.
Let me begin. Yeah. I'm Ambrish Kumar. I'm passed out student from SCIS JNU, and my designation is currently MTech. I'm not PhD, so preferred is not to call me Dr. Ambrish. So I'm starting my presentation. My topic is workflows for genomic variants detection, and I have worked over three workflow management systems, and I'm starting from the introduction. This is my first PPT. SOPs and workflows. Standard operating protocols are a step towards standardizing genomic variants discovery. Workflows and workflow management systems are adding reproducibility and reusability to the bioinformatics research. We have formed, in fact, I
Yeah,
have formed three standard. Yeah.
sorry to interrupt you. Please, could you go for full screen?
Yes, sir.
Yeah, full screen, please.
Is it okay, sir?
No.
No, you have to make it full screen because right now you are only opening the file.
Okay, sir.
Yeah, please press F5. F5. Thank you.
Continue, please. It doesn't. It's okay. You can see.
Is it okay?
Yeah, it's okay. Please continue.
Thank you. Thank you so much, sir. I have formed three standard operating protocols for genomic variant detections. Used tools for the variant detection are SAM tools, VARSCAN, and GATK. Separate SOPs for each tools are present, and we have converted those SOPs into workflows. Used workflow management systems to form workflows are Workflow Description Language, WDL, Common Workflow Language, CWL, and Galaxy. Implementation steps are formation of WDL scripts and CWL scripts and formation of Galaxy workflows. We have formed separate Docker-based and non-Docker-based WDL scripts for each SOPs, and we have executed each WDL scripts over the Chromewell execution engine, which supports Docker as well as non-Docker runtime environment. CWL tool for each step of SOPs are developed and connected together for the semantic execution as a workflow in a single CWL workflow script. CWL runner is the execution environment, provides the execution environment to the CWL workflow. And we have formed Galaxy workflow for each SOPs. We have available Galaxy tools for each step of the SOPs, and connecting them, we have formed the Galaxy workflows. And here is the next implementation. Workflow is the execution engine for the WDL scripts. CWL runner is used for the execution of CWL workflows, and Galaxy provides interactivity to end users to run their tools and Galaxy workflows. Again, we want to mention that WDL workflows are run into two modes: command line as well as server mode. CWL workflows are run into two modes, that is command line modes as well as its runtime platform, that is Arvados. More than one platform support the CWL execution, but I have currently used Arvados platform, so I'm mentioning only two mode of CWL workflows execution, that is command line and Arvados execution. And Galaxy has the only way to execute the workflows is the using user interactive interface, that is the GUI of the Galaxy. And for the performance benchmarking of each tool of the genomic variant detection, we have generated manually mutated Ebola reference genome containing 10 SNPs, 10 indels, 5 inversions, and 5 translocations. Paired and FastQ Ebola RNA-seq reads are simulated using RSAM, and this dataset is submitted to NCBI SRA database as a bioproject. Here is the summarized SNP detection performance for each tool. GATK has sensitivity of 0.8 for the SNP detection, precision of 0.77. VARSCAN has SNP detection sensitivity of 0.9, means 9 out of 10 SNPs are detected by the VARSCAN. And precision is slightly low for the VARScan now, because I use the default parameter setting for the VARScan. It pick mutations from translocations and inversion regions. Its precision is 0.13. SAM tool has SNP detection performance sensitivity of 0.8 and precision of 0.44. Indel detection performance for the GATK is its sensitivity 0.7, precision 0.7, VARScan 0.4, precision 0.57, SAM tools 0.7, and precision 0.41. SAM tool indel detection is very powerful in terms of in comparison with other two, because it detected almost all SNPs which were called into the Ebola reference genome. Although GATK has equal sensitivity with the SAM tools, but its result, there would be some redundancies picked by the GATK. So I will emphasize that indel detection of SAM tool is much better than even GATK and VARScan. Here is the timing diagram of Cromwell execution, which executed GATK workflow for SNPs and indel detection. And here are the this is the calibrated seconds which each step has taken to execute. This is the haplotype caller. This is haplotype caller execution time. This is the variant. This is the next step of haplotype caller. Next is, this is the execution of GATK tool for executed into CWL workflow for SNP and indel detection using GATK. This is the execution diagram generated by CWL runner. These are the schematic. This is the schematic diagram for each step of GATK workflow, which are involved into CWL workflow for GATK SNP indel detection. This is the snapshot of Galaxy workflow for the SNP and indel detection using GATK. Alignment, this is quality control, pre-processing of the data, duplicate marking as a GATK for mutate 2 caller, variant filtration, annotation, and concatenation of different variants from different samples, all variants from the different samples. This is the initial step which takes the multiple input at a single time and concatenates all SNPs and indels into single file. Conclusion is that overall performance in terms of sensitivity and precision of GATK is better than others too. VARS can pick mutations from inversions and translocations regions. SAM tools shows better performance in indels detection. Future work remains to enhance the usability and scalability of the workflow to high-performance computing platforms, as well as cloud infrastructure to enhance the usability and efficacy of the research. This is the link for submitted SOPs and workflows. One can easily access the SOPs and all the workflows, CW and Galaxy workflows submitted to Workflow Hub. I had contributed it as a biohackathon project, and my contribution is already acknowledged on the Workflow Hub EU. And these are the use references which I have used while the study, forming the manuscript and making the presentations. This is the dataset which is hosted at Genodo. This is the DOI for the test dataset. One can easily download it there and used on the Galaxy platform and use it as a toy dataset. And next, I acknowledge Galaxy Europe, CWL, and Arvados for providing computational infrastructure support and guidance for forming the workflows. I got much support from the CWL main person and the Galaxy Europe. They provided much guidance for making CWL tools and executing them into single workflow script. Thank you all for the patiently listening the presentation. And I would like to invite questions from your side.
Thank you, Amrish.
Thank
We
you.
can take 1 or 2 questions if you
Yeah.
any. Maybe Agam Mishra can start preparing for his uploading of the presentation if in the meantime. No questions? Okay. I thank you, Amrish. Nice presentation, and you have evaluated three different platforms and the workflows.
Yes, sir.
So, may I now invite Mr. Agam Mishra, who would be talking on computational investigations to analyze genes involved in lung cancer association repair mechanisms. Dr. Agam, is he there? Hello? Hello? Is he there?
Yes, sir.
Yeah,
Yeah.
please.
Sir, is she, sir? Yeah.
Okay, I'm sorry. Uh, is she there? Is she ready to present?
Yes, sir.
Yeah, please.
Just
Make it presentation mode. You're still.
good afternoon. Good afternoon, everyone. Is my presentation visible?
Yeah, it is visible. Could you make it in the presentation mode? It is still.
Oh, 1 minute.
Yeah, that's right. Thank you.
Okay. So, my name is Aga Mishra. I'm a fourth year BTech student at the Department of Biotechnology and Bioinformatics at JP University of Information Technology, Solon. I'm going to discuss on the computational investigation to analyze genes involved in lung cancer associated repair mechanisms. So, lung cancer is the leading cause of cancer related mortality. According to WHO, there is 135,720 deaths that occurred due to tobacco consumptions and cigarette smoking. Development of this lung cancer involves loss of genetic stability, which results in increased genetic changes and mutations. Here is the statistics, which shows the estimated number of new cases in 2018 worldwide for both the sexes. Here we see that there is 11.6% of cases that are associated with the lung cancer, followed by the breast cancer and other types of such cancers. In order to cope with genomic stresses, the cell employs a diverse array of 160 DNA repair proteins. There are 5 distinct major DNA repair pathways in human cells: base excision repair, nucleotide excision repair, mismatch repair, non homologous end joining, and homology directed repair. This study addresses the association between the lung cancer and genetic instability, with a central focus on genetic mutation in the DNA damage repair pathways, that is, the biomarker staging and direct therapeutic targeting. We took our data from the gene expression omnibus. It was basically the microarray data, and then we analyzed our data using the computational analysis through various databases and pipelines such as the WBDEGS, GEO2R, and DEVID. With the help of WBDEGS, we found this graph between the stepwise linear regression and the significance. Here we can see that the values, that the genes which are above the zero point 0.5 value are the insignificant genes, and these are the experimental noise which are needed to be removed. And the value and the genes which are below zero point 0.5 are the significant genes. Further, we analyzed it through GEO2R, where we found that most significant genes are with smallest p values. And here in this graph, the red bar represents the expression measurement that is calculated through the value column presented in the sample records. After, with the help of David, we did various functional annotations and clusterings and derived the biocarta pathway. With the help of gene expression data, we predict genes such as BRCA1, BRCA2, CHK1, and ATR, which play important role in chemotherapies in non-small cell lung cancer patients, as well as they act as the target biomarker in identification and control of disease. As a result, BRCA1 gene act as the promising genomic marker and can be used for customized chemotherapy in non-small cell lung cancer patients. Non-small cell lung cancer patients shows longer survival with negative expression of BRCA1 or CHK2 as compared to the presence of positive expression of such genes. CHK2 has the prognostic significance and it is best predictive factor in non-small cell lung cancer. ATR causes delay in cell cycle by activation of CHK1, which gives time for repairing of DSBs. This pathway best explains how BRCA1, BRCA2, and ATR promotes hypermethylation in 2.56% of non-small cell lung cancer patients, which is described as the early event in lung cancer carcinogenesis. Not only this, it also acts as the modulator of chemotherapy response in such lung cancer patients. Hence, I would like to conclude that uncontrolled cell growth in tissues of lungs led to the development of two major types of lung cancers, that is the non-small cell lung cancer and small cell lung cancer. Analysis is done on the gene expression data to predict the novel genes such as BRCA1 and BRCA2, which can act as the biomarker or disease control target for identification or control of disease. These genes will provide the pathway level information when we will use it for the system biology approach for the network analysis. Additionally, sequence and structural level analysis of genes for SNPs and their impact will help the experimental biologists to look for possible direct targets conditioned to their experimental validations. These are some of the references which I looked upon. Thank you.
Thank you, Agam. Nice talk within the time limits. We can take one question before I ask Satya et al. That is presented by Nidhi Shukla. We can take one or two questions if there are any. So if there are no questions, then I'll would like to request the poster talk by Satya et al. It is being presented by Nidhi Shukla. She's at BISR at this place only. And that is, the title is Prioritizing the Regulatory Elements Associated with Prostate Cancer Genes. Nidhi.
Thank you, KM sir. Good afternoon, everyone. My name is Nidhi Shukla. I'm working at BISR. So I'm going to give a poster presentation titled Prioritizing the Regulatory Elements Associated with Prostate Cancer Genes. So before moving ahead, I would like to give a brief introduction so that everyone stays on the same page. Prostate cancer is the second most common cancer among men, and initially, it was thought that it's not that common in India, but recent population based cancer registries have reported incidences from almost all parts of the country. Long non-coding RNAs are the ones which are more than 200 nucleotides, and they doesn't code for any protein. Initially, they were thought to be as a junk DNA or noise DNA, but they have gained like worldwide attention because they play many regulatory roles in different diseases, and cancer is among one of them. So the aim of our project is that we want to identify like causal genes and mutations specific to PCA in Indian cohort. And as far as we are aware, there are not many NGS studies has been done on prostate cancer. With this aim, we also want to identify regulatory elements associated with these causal genes. So for that purpose, the methodology what we have followed is shown here in a flow chart. So we did a whole exome sequencing of 10 samples where like 5 was cancerous and 5 was control. And by the advent of this, we were able to identify 30 causal genes. Uh, then we blasted these genes against non-code database, which is a repository for long non-coding RNAs. Out of that, we got 12 hits, And then lastly, like we shortlisted 5 long non-coding RNA based on some criterias which are mentioned at the left side. That is their expression in prostate cancer, their interaction with other PCA causal proteins, and if they are known to be involved in different cancer pathways. Simultaneously, we were also interested to identify any regulatory elements associated with this. So we used a genome walking approach in by using like Ensemble database, where we were trying to move like upstream of these genes, and we were looking for enhancers, CTCF motifs, or open chromatin elements. So if I talk about the result, so this panel, it shows the list of 30 causal genes, which is mentioned here. And as you can see, there are many important genes which we found out, like BRCA1 and BRCA2, which are well-known mutations in breast cancer, and they have also been known to be reported in prostate cancer as well, along with P53, ADA. So we are in process of validating those genes and trying to find the mechanism, like how they are regulating prostate cancer pathogenesis. So the right side table shows the long non-coding RNAs and their interaction with different causal proteins. So we are currently we are validating its validating these long non-coding RNA and doing some quantification study using real-time PCRs. The lower side, this table, it shows some this regulatory elements which I mentioned earlier. There's some CTCF ID and enhancer ID. I'm showing this only for BRCA1, but we have finished like almost like all these genes. So just to conclude this part, that so far we have been able to identify numerous regulatory elements such as enhancers, OCE and CTCFs, which needs to be further validated. And all these studies will give a hint of like PCA pathogenesis, and that can be involved in identification of biomarkers. So this is all I have right now, and I would like to thank Dr. Prashant for his continuous encouragement and guidance. Thank you so much, and I would be happy to take any questions.
Thank you, Nidhi. If there are any questions, please. If there are no questions, then we will open the floor to, you can ask any question to any of the speakers in this session. Devasis, Abhishek, Prashant, Ambrish, Agam, anyone, Nidhi. I have one question to Devasis. Are you there?
Yes, yeah. Go ahead.
Yeah. So my question was, I mean, have you also given the input of the metagenomic data or only the pure culture data?
No, this is the only the whole genome data is given.
Pure isolate.
The whichever microbiota, the whole genome is there. This was actually based on another study which had come in Nature. I think it is probably Nature Biotechnology or one of the journals. So around 1500 with the gut microbiota, they had the whole genome sequences. So that is the one which I showed in that bar plot slide. But in that work, they can only classify as the broad pathway, that is a polypeptide synthase pathway, non-ribosomal peptide pathway, or things like that. So what this machine learning and our bioinformatics tool, RipMiner Genome or SBS-PKS is doing is, they are able to predict the chemical structure of the final secondary metabolite.
Okay, thank you. Thank you. I have another question to Dr. Prashant. Could you tell me how much amount of the serum or plasma that you require to? I mean, do you find any problems in the isolating the cell-free DNA? Is it there? Dr. Prashant? No.
Sir, I think he's not there.
He's It's not
Prashant,
there.
you need to unmute if you're talking.
Yeah, sorry. Sorry, sir. Prashant is not here, but maybe Abhishek could answer. Abhishek, is Prashant around? Yeah.
I do not see him actually. So I will ask the question and I'll respond to you later. Yeah. So
Yeah.
yeah. So it will be a specific question to him. I will raise the question and get you answer by email, sir.
Sure. Thank you. If there are no more questions, I'll thank all the speakers in this session for their excellent talks, and also to the audience who are patiently listening to this and actively participating in the discussion. Back to the organizers.
Yeah. Thank you so much, KM sir, for wonderfully chairing the session. This has been immensely helpful, and your wonderful insights are always helpful. So next we have a session on plant and elite genomics that Professor P.K. Gupta and Dr. Vijay Gailot will start in about 12 minutes time. So until then, please have a quick break and I'm at this busy schedule. Thank you so much. PK Gupta sir will be starting in about 12 minutes. Until then, please enjoy this background music. Yeah.
हाँ भाई, तुम्हारे से जल्दी ना देखना पड़ेगा। नहीं, अभी तो मेरे पास तो ये 90% है लाल से।
ji, namaste. This is Rajiv.
Hello,
Yes, sir.
Rajiv, how are you?
I'm fine, sir.
Prashant, can we start?
Yes,
Or should we
we
wait?
take honor in inviting Professor P.K. Gupta and Dr. Vijay Gailot, who needs no mention. P.K. Gupta sir was already introduced. Vijay Gailot is a senior scientist at CSIR IHBT Palampur. So we take honor and pleasure in having P.K. Gupta sir for this chairing this particular session. So over to you, sir. Thank you so much.
Okay, Prashant, thank you very much. We have three invited talks in this particular session and four oral presentations. Most of them are young scientists, brilliant young scientists, and I've seen their biodata, excellent biodata. Dr. Girish Mohannath is an assistant professor in the Department of Biological Sciences, Pilani, Hyderabad campus. He has been the recipient of Ramanujan Fellowship of DSC and also Ramalingam Swami Fellowship of Department of Biotechnology, and his interest is epigenetic regulation of ribosomal RNA. Great pleasure for me to invite him to deliver the lecture. Vijay Gailot will take over after the first presentation. Is he there? Dr.
Yes, sir.
Mohannath? Dr. Mohannath is there?
Yes, sir. Is there, sir? Girish?
Has to unmute.
Is he there?
Yes, sir. He's there, sir. Maybe he's unmuting his microphone. Girish?
Okay.
Yeah.
Some problem?
Girish, if you're around, please start your presentation, please.
Yeah.
You have already been introduced. Thank you so much.
Yeah.
Okay, thank you. Sorry, there was some glitch. Okay, I will start. No. Can you guys see the slides?
Yes.
Okay. Thank you, sir. Thank you for the kind introduction.
Welcome, young scientist.
Sir, under your guidance, sir.
Yes. Good luck to you.
Thank you, sir. Thanks, Prash. Thanks for the organizers. Let me outset tell you that I am basically a biologist, but with the several help of several bioinformaticians like Prash and others. We have been trying to answer some interesting questions, which I will share in the next 20 minutes.
Make it full screen.
Yes, sir. Yeah.
Yeah. Okay.
So the title says bioinformatic analysis of 45 RNA genes from plants and humans. So we have analyzed from plants and used it to answer some questions. It has been published, but humans needs to be done. Certainly our lab alone cannot do. We were initiating some efforts, but I am going to give you, those of you who are interested to analyze these sequences, quite a lot to be done for humans. Okay. But just to let you know, our lab is more interested in rRNA gene regulation, not from the, looking at the for the identification of species in that context. Okay. A brief introduction. These genes are clustered at chromosomal locations called nucleolus organizer regions. Most of you might have heard this term in your biology courses. They're called knots. If you just look at this diagram, a chromosome I have shown, and here is a NOR region, this is in Arabidopsis, for example, but it's pretty similar in all eukaryotes. These genes are all organized as tandem repeats, okay, and they code for this ribosomal RNA components, which become part of ribosomes and for protein synthesis, okay, which we all know from basic biology. If I can just quickly show you, kind of an animated slide here. Our RNA polymerase 1 transcribes and then gets processed, and these RNA becomes part of ribosomes as large and small subunit, and they are perform photosynthesis, and they are in fact ribozymes actually, because rRNA is what provides the function of the catalytic part. So the number vary from less than 100 to more than 10,000. And 5S rRNA genes are transcribed by RNA polymerase 3, whereas 45S rRNA genes are transcribed by RNA polymerase 1. As you if any of you have loaded RNA on gel, you would see what you see as a band is usually rRNA or tRNA. Okay, so that clearly tells that's the most abundant species in the cell. And if you are wondering, why we study RNA gene regulation, there are a few reasons. One is the scale of gene silencing is pretty large. We are talking about megabase scale silencing. It is next only to X chromosome inactivation in mammals. But it is not random or due to maternal or paternal effects, means there are some genetic mechanisms involved in gene silencing. So we would like to uncover those mechanisms in the long run. And the misregulated RNA gene synthesis and instability are linked to several human diseases and mainly cancers. Okay, a part of that we are doing in our lab. And because of these genes are upregulated, they have been already used as anti-cancer therapeutic strategies. So I will later on again touch upon this again later. And this involves mechanisms of epigenetic regulation. Sorry, it involves epigenetic chromatin modifications. So rRNA genes serves as excellent models to study the effects of chromatin modifications because they are regulated by these modifications. And some of the recent papers suggest that the DNA alone appears to play in heterochromatin formation. So how it is, needs to be addressed. And very recently, someone showed in a genome research paper that it harbors an evolutionarily conserved clock of biological aging. I think as it gets aged, it gets methylated, this DNA. I think it's linked to aging in multiple animal models they have shown. So if you look at humans, there are 5 knots on 5 different chromosomes. Each gene is about 40 kb length, including intergenic sequences, spacers or intergenic spacers. This paper says there are around 300 copies, some other says 450 to 600. So the number seems to be varying from individuals to individual, or is it just, still how accurate estimate, we don't know, but somewhere between 300 to 600 copies. So if you look at the human genome size, is about 3 gigabase, and about close to 0.8% of that genome is ribosomal RNA genes. Whereas in plants, we study Arabidopsis thaliana. Okay, so there are 2 knots with 400 copies each. Genome size is 135 megabase only. So it's about 6% of the genome is ribosomal RNA genes here. Okay, but unlike humans and other mammals, each gene is about 10 kb long, including the intergenic spacers. Okay, the intergenic spacers are pretty long in humans. About 30 kb constitutes only intergenic spacers, but in plants it's only about 2 kb. Okay, so now when you look at these genes, they're all very conserved. Okay, and although eukaryotes may have lots of genes, only about 50% of them are get expressed, remaining are selectively silenced. That was one of the questions we answered earlier. How do they get silenced? Okay, so when we looked at these genes, they're pretty much conserved across those genes, but genic regions. But there's one particular region, this 3' ETS, externally transcribed sequence region. It is variable. If you design primers here and amplify, you will get 4 variants, of which only one is expressed. As you can see on the right side, if you see the expression data, the variant 1 is silenced. All these are based on length polymorphism. But however, the question was, how are these targeted for silencing? Okay. And then the previous hypothesis, maybe each gene is targeted individually on different NORs. But accumulating a lot of evidence, we showed that it is not so. It is rather chromosome based. On chromosome 2, they're all silenced. On chromosome 4, they're all active. Okay. So this was the question we are trying to address. In this context, I want to tell you how we used bioinformatic analysis to answer this question, a part of it. Okay. So we provided multiple lines of evidence. I will just mention one line of evidence where we use bioinformatic analysis. So one is, of course, these are length-based, polymorphism-based variants. These can be mapped using standard mapping approaches, marker-based mapping approaches. We showed that all the active variants map to NOR4 and all the inactive variants map to NOR2. Okay. But these are all, based on only length polymorphisms from the 3' ETS region. So we thought that is not enough. So we used the deep sequencing analysis to identify SNPs. And there were about 15 SNPs were identified all along the genes. Okay. And the ones which are shown in green means those are the active SNPs. Those which are shown in red is the silenced SNPs. Okay. So once you know a SNP, it can be used to generate a marker, either CAPS marker or DCAPS marker, and you can also analyze their expression status. Okay. So here quickly I will show you a CAPS marker based assay if you don't know, if you are not familiar with this. Let's say if there is a one polymorphism here, just a single nucleotide. As you can see, instead of T, here you have G. Okay. So this itself creates a MSC1 site in one case on the right side. On the left side, this is no MSC1 site. You can amplify using a primer this region and get amplicons in both the individuals. The polymorphism comes when you digest with that MSC1. So in one it gets digested, in other it doesn't. So this is a classical CAPS marker assay. These markers can be used for multiple reasons, multiple purposes. Mainly it can be used for mapping. Okay. So same thing you can do for a mapping population. You have homozygous for one or the other, or it can be heterozygous. For example, if you see 3 bands, means it is heterozygous. All right. So once you know a SNP, you can analyze their expression status. You can say whether the SNP is expressed or not. this is a panel of some SNPs we analyzed. I will just show you one quick example here, which I am showing with a blue square. You see at the DNA level on the leftmost lane, please pay attention only to the one which I am shown with a blue box. On the leftmost lane, you see the particular amplicon without digestion. Once you digest, only a little bit get digested at the DNA level. Means whatever that gets digested carry a SNP of T in that position. If it does not get digested, those genes have A in that particular positions. But when you see at the DNA level and compared to the RNA level, you can see most of it gets expressed. Means most of it gets digested. Means the genes which have T in that position, that particular SNP variant is expressed, whereas the other one is silenced. Okay, relatively. Like that, you can analyze for all the SNPs and you can also map. However, Picard Lab has recently shared update with that is Craig Picard Lab is where I worked as postdoctoral researcher before I left US. They have sequenced using Oxford nanopore technology, and they have identified more variation, more variants, and which is under the right now process of getting published. But they have shared some of the raw data for some of our analysis, which briefly I will show you in the next few slides. So whatever our findings were, we published in this journal, and it got highlighted. Because we proposed a new hypothesis that these ribosomal RNA genes are regulated based on chromosomal location, not based on sequence. But bioinformatic analysis were very useful to identify SNPs and their mapping and study their expression status. Now, while I was completing, about this work and the questions we started asking, because I was planning to move to India. What about in humans? Whenever we present this work, we would ask, okay, what happens in humans? Well, there are 5 NORs in humans. Is it also NOR-based regulation? Let's say maybe the chromosome 13, 14, 15 are on and the remaining chromosomes are off. Or how does it regulate? Well, the answer is not known. People have not sequenced these sequences extensively, and the answer is not known. I kind of just tried to propose as part of my Ramanujan Fellowship application 4 years ago, and maybe I'll try to initiate some work in that direction. So there is a lot of relevance for this gene regulation in humans, because in most of the cancers so far they have studied, they all get upregulated. Okay. Interestingly, in some cases, this upregulation of these genes is linked to rDNA promoter hypomethylation. In some cases, it is not. Now you can link, I have been told that recently that nanopore technology sequencing data can also give you information on the methylation status. Okay, so if someone can combine the methylation status and sequence these genomes, these genes specifically, I think it can tell you the difference between the active genes and inactive genes. Are they based on chromosomal locations, or are they how they are specifically regulated in cancers? One can be obtained. So, and there are also instabilities in cancers. These are hotspots for cancers. Okay, and because they are upregulated, some groups asked, what if we downregulate these ribosomal RNA genes in cancer conditions? Can we control cancers? So the answer turned out to be yes. Ross Annan group in Australia and Laiho group in Finland. Of course, she has a lab in US too. These two groups are predominant players in coming up with different compounds which which inhibit RNA polymerase transcription and serve as anti-cancer activities. And the first one is actually in the clinical stage trial 3. I think clinical stage 3 recently we updated ourselves what's going on, so it was encouraging to see that it's been okay. Now, with the help of Prash and other bioinformaticians, we made some efforts in the beginning to see can we analyze the existing human genome sequences, because to analyze ourselves whole genome is a tricky, means it's expensive because it's only less than 1% of the genome. It's like a overkill, just to get 1% of the genome. You don't want to go to the sequence the whole genome. We were about to initiate some 20 individuals, sequencing, targeted amplicon sequencing, but then this COVID-19 came, and now, drawing blood from individuals is a lot more riskier. So we have to put that on a shelf. We have applied for a project. We'll see if we can proceed further. But in the meantime, you guys aware of this 1000 genome project where genome sequences are available. Although the depth is very less, still one can make use of it. And we contacted this guy called Dr. Artem Babayan, and he has analyzed these genome sequences, and he found that there is a intra and inter-individual genetic variation exists in RNA genes. Okay, and morning also you heard about Genome India, where is about 10,000 individuals will be sequenced, whole genome sequence for the Indian individuals will be available, and I think that will serve as an excellent source resources to analyze RNA genes from Indian population. So far, there is no reference sequence available for Indians yet. Okay, so that can be also done, and some work can be initiated. So another aspect I want to quickly highlight is while working with Arabidopsis, we observed that there is a non-homologous recombination happens in these mutants, some of the mutants. Okay, so how could we know that? Because we sequence the subtelomeric regions which are unique, and there is a technique to design primers. It's a little bit tricky. We can, through amplification, we can tell if these junctions are there or being deleted. For example, here I'm showing a mutated, it's get deleted. Then another ecotype, I wanted to clone those junctions. I am not able to do it. Then we thought, why don't we make use of the bioinformatic analysis? There are sequences available we wanted to identify. Unfortunately, we found that these non-telomere junction sequences are poorly represented across different sequencing platforms. 3 groups worked on it. Prash, his group tried, a couple of other friends in University of Hyderabad, and then Picard Lab tried using different platforms. The idea is like that, we don't know this junction sequence. We tried to clone it. It was like earlier strategies. We could not. Then we thought, can we get these sequencing reads which overlap with both the telomere and the NAR sequences, so that then you can get exactly the junction sequences, and then you can use that to clone and further verify it. So this was the idea. We tried using the NGS data available in the 1000 genome projects for Arabidopsis. We were not successful. And recently published PacBio sequencing data, which was, we used, we did not. And luckily, the Oxford Nanopore technology platform data, which Picard Lab has obtained and yet to be published, they found some reads coming from this region. So Dallin Fultz is a postdoctoral fellow there. So they give these reads and assembled it and gave us the potential sequence coming from these sections. As you can see, this is probably the case with all the organisms who are trying to get the subtelomeric sequences. It's not easily represented. See, if you're talking about the NGS and particularly where some adapters are involved, it's understandable because telomeres are repeats. To clone those junctions is difficult. But nanopore should run through the every DNA. But why were not in PacBio sequencing kind of platforms? These junctions were poorly represented. Particularly those who are involved in sequencing may pay attention. See how to improve the coverage, particularly to the end regions. And so we got some reads. Based on that, we are trying to clone these junctions. Again, the question is, has anybody cloned these junction sequences in human chromosomes? Certainly not. I think they are not done yet, as far as I know, unless if it is very recently published. At least few years ago, I know that it was not. So again, there are some repeats, and it's not much efforts have gone in that direction, but it will be useful because what if they are recombining with each other? You can tell that how frequently they are recombining from one chromosome to the other chromosome. Okay? And lastly, in 1 or 2 minutes, I will just quickly tell. We all talk about, genome sequences. It has been sequenced. We know the genome sequence, but how reliable these genome assemblies are. Okay. I think there are still lots of gaps exists in many genomes. I think we keep on. I think correction will go on for at least another couple of decades. It looks like. And I will share one example with some data. So we try to analyze just 1 megabase region adjacent to